In this segment, we will discuss how DNA methylation patterns they change in the cancer cell. Because the cancer cell is also a very important model to be discussed here. And the DNA methylation, because it's silencing certain signals there, how it is there. Now, DNA methylation, as we study, is one of the most important epigenetic alteration and plays a critical functional role in the cell life. Because in the epigenetic system, it is there, it changes the non-sequence message to the body or to the cell. And what happened that the DNA methylation, it is in epigenetic alteration, what the role it plays. And it plays the role in three important areas. One is the development of the cell. Second is the differentiation of the cell. And the third is the diseases which the cell is going to be prone to. जो कि cell diseases के साथ क्या करेगा, क्या सलूक करेगा. Now the pattern of DNA methylation, it changes. ये एक तरह नहीं रहता. Different जब environment होते हैं तो it changes. And it changes substantially, काफ़ी हद तक, when cell become cancerous. जब cell एक cancer बन जाता है, cancerous हो जाता है, तो the pattern of DNA methylation, it changes. Now what is a cancer cell? Cancer cell, as a matter of fact, is a cell which has lost the control on its growth. इसे नहीं पता कि इसे कब मरना है, इसे नहीं पता क्या करना है, इसका यही है कि it has lost the control and it is going to grow. बिल्कुल ऐसे ही जिस तरह if you have a car and you have a broken throttle and no brakes, so the car will keep on going. Now, there are two major phenomena which are related to the change of pattern in the methylation of a cancer cell. In the first phenomena, what happened? The tumoral genome become globally hypomethylated. ये methylated हो जाता है. Unlike in normal cells, mainly due to generalized demethylation. एक तो generalized demethylation हो जाती है ना, लेकिन इसमें क्या हो जाता है? It becomes hypomethylated. And it is the generalized demethylation in the CpGs scattered throughout the body of the genes. CpGs जो हैं वो बॉडी के जो सेल है उसमें जगह जगह बिखरे हुए हैं और इस फिनोमेना को ये करते हैं. Now the second phenomena वो क्या है? It is local and discrete region situated at the promoter region. जो promoter region है उसके ऊपर ये लगे हुए हैं. And the promoter region of tumor suppressor genes, it undergoes intense hypermethylation. ये intense hypermethylation में promoter region में जो है ये टेक ओवर होता है, उनके ऊपर इसका इफेक्ट होता है. For example, see this. That's a normal cell on the top. And there are promoters, there are this, and transcription is there. So there are control on all that. But what happened in the bottom? There is a malignant transformation. That is a cancer cell. Remember, in the cancer cell, there is a change. There is a different methyl group. They are removed. Then further methylate are there. And the gene silencing occurs eventually because that gene is not being expressed. And What happens when that gene is not expressed? The cell is out of control and it keeps on growing or doing that. Now, global DNA hypomethylation in human cancer, what is its function? And the first response actually linking the aberrant DNA methylation to cancer came from Lepre and Becker. These are the two scientists who determined the 5-methylcytosine content of the normal cell in liver and hepatoocular carcinomas, जो कि कैंसर सेल्स हैं, induced in rats by acetyl aminofluorine or diethyl nitrosamine. दो कंपाउंड है जिनकी वजह से उनने उसमें ये इंट्रोड्यूस किए. And carcinogen induced cancers, that is displayed a decrease in overall genomic modification. रिजल्ट क्या हुआ कि कार्सिनोजेनिक जो कार्सिनो थे, जो कैंसरस थे, इंड्यूस कैंसर, ये डिक्रीज हुआ इन ओवरऑल जीनोमिक मैथिलेशन about 20 to 40%. 20 to 40% that was decreased, and relative to normal river, जो normal river था उसके relatively 20 to 40% that was decreased. So what does indicated that this is an early event in cancer development and it accumulates. जब early event है तो उसमें क्या होता है? It accumulates throughout all tumorogenic steps. What happened from the beginning proliferation to the invasive cancer? पहले उसकी proliferation जबकि initially वो grow करता है, and then to the invasive cancer, that when it goes to a cancer disease, it does that. And the gene-specific methylation, it occurs in the content of global DNA. Global DNA hypomethylation में क्या हो जाता है कि gene-specific demethylation हो जाती है, and DNA के content में hypomethylation takes place. Many of the effects, they arise through the activation of transposable elements and endogenous retroviruses. ये किस किस के थ्रू हो जाती है कि ट्रांसजेनिक जो हैं वो ट्रांसपोजेबल एलिमेंट्स, सिमिलरली एंडोजेनस